Welcome all to Life Science Academy. Today we will learn about the DNA replication, how the DNA get replicate. So in this session we only focus on the DNA replication process in prokaryotes. So what is DNA replication? DNA replication is the process by which a DNA molecule get duplicated, or you can say replicated, or you can say making an exact copy of their genome. So in this session we will learn how the prokaryotic cell, or you can say organism, replicate their genome. So the prokaryotic model organism is the, as you all know, that the E. coli. E. coli is the model organism in the prokaryotes. So before going to detail in the E. coli genome replication, we have some prospects in the molecular biology, and this topic I have taken from the molecular biology. So these central dogma of the molecular biologies are: the first central dogma is the replication, second is the transcription, and third one is the translation. So if you want to summarize all the molecular biology, so this summarize around these three prospects, and these prospects are the fundamentals of the molecular biology. So let's discuss one by one. In this session we will learn how the DNA replicates its genome in the E. coli. E. coli is the model organism. So if we talk about central dogma of molecular biology, so if we have DNA and this DNA get replicate and making exact copy of this DNA, this process is called the DNA replication. In another word, we can define the DNA replication: the genetic material DNA get replicated with exact of their parental genome. This process is known as the DNA replication. In another word, you can say making of DNA from the DNA as a template is called the DNA replication. So if you have DNA molecule and you want to make the DNA molecule, you must have a DNA as a template. So you can replicate the genome. So these central dogmas are important in the molecular biology. So this is the process in which DNA to DNA, making DNA to DNA and DNA to mRNA, and DNA to mRNA is called the transcription process, using DNA as a template strand, and making of protein using mRNA as a template strand is called the translation. And this is the protein which have functional role in all the organism and all the significance in the molecular biology. A functional protein can work as a modulator or regulator of all the functions in the cell. So if we talk about semi-conservative model of DNA replication, the semi-conservative model of DNA replication said that if we have parental DNA and this DNA want to replicate, so this DNA could be unwind. After that a new DNA, let me change the color, a new DNA get replicate. So this new DNA have the parental as well as new characteristics. As you all know that in human we have 46 chromosomes, 23 coming from each parent, or you can say 23 pair of chromosome. So if the two DNA molecule get recombinant, we have semi-conservative model of the DNA replication, and this model is the well accepted model by the Watson Crick. So in another word, DNA replication is the process of making duplicate copy of DNA using DNA as a template strand. We must have template for replicating the DNA strand, which is exact copy of parental DNA strand. This process follows these 3 steps of replication, and these 3 steps are the initiation, elongation and termination. So let's forward first initiation process in prokaryotes, or simply the E. coli. E. coli is the model organisms. So what we want to requirements for the initiation of the DNA. So we have some enzymatic machinery required for the process of initiation. These enzymatic machineries are the DNA A, DNA B, DNA C also, and SSBP, DNA guidase, DNA G and DNA polymerase. DNA polymerase is the measured replicating enzyme in the prokaryotes. So how the initiation starts in the E. coli? We have a circular genome of E. coli. If the E. coli wants to replicate its genome, if we want to one E. coli from a single E. coli, so we have, I want to replicate first its DNA. I want to first replicate its DNA. And this DNA is exact copy of parental DNA. Exact copy of parental DNA. This is daughter. So in prokaryote there will be the cloning process. There will be the clone. So no changes occur in the cloning method. So let's start the initiation process in detail. So selection of replicator site. So as you all know that in E. coli, this is the circular genome. This will be the circular genome, and this circular genome where the selection or where the ORIC present. ORIC is the origin of replication, and this ORIC site decides the initiation of replication. And this ORIC is approximate to 45 base pair long in E. coli. Escherichia coli, and in this 245 base pair, between them there will be some prospects, and these prospects are the 80 rich sequences. 80 rich sequences which will responsible for the easy melting and unwinding of the DNA. So there will be 3 30-mer sequences and 9 mer, 4 to 5 9-mers are present in the ORIC region. So this is the site where the initiation gets started or could be started. But when the DNA get replicated, this will decides the enzymatic machinery of the E. coli cell. And I have told you previously there will be some enzymatic machinery which will require to initiate the replication in the E. coli, or you can say in the prokaryotic cell. So first you have to load, or you have to add the DNA A, which is the initiator in the replicator site, or you can say replicator site. After that, DNA C comes. Before going to replication, there will be a formation of some initial melting occur. Why this occur? Because the binding of the DNA A protein in around the 80 rich sequences, or in the region where the 80 rich sequences are present, and this 80 rich sequences are defined the ORIC region in the prokaryotic cell or E. coli. After that, loading of DNA B. And what is the DNA B? DNA B is the helicase, and helicase is a hexameric protein, and it have ATP hydrolysis activity, and DNA A also have ATP hydrolyzing activity. So if we have mechanisms, so we can easily understand the initiation process. So let's start. So we have the, let me change the color so you can, okay, let's start. So if we have the DNA, this will be the DNA, and if the DNA A protein comes, what will do? D, and there will be some regions. or you can say specifically this is the ORIC. So DNA comes. So DNA comes. So this is the DNA A protein. So this is the DNA A protein, and this will be the some melting in the AT rich sequences. This melting make an initial melting to facilitate the another enzymes. After that, there will be the DNA A and C. C is a helicase loader. So, in another word, you can say DNA C comes with the DNA B, and DNA B is a helicase, and helicase activity require the ATP hydro. This is ATP hydrolysis required. DNA A also require ATP hydrolysis. You must know where the ATP require, where the ATP not require. Okay. So after that, DNA B and DNA C comes with. After that, there will be SSBP protein, single stranded binding protein. This single strand binding protein, what to do? He is due to prevent the reassembly of melted DNA. After that single stranded binding protein, you have like this scenario. Change. You have a picture like this. So this is the hexameric protein, and this hexameric protein is DNA B, or you can say helicase, and there will be the binding of tetrameric protein, which prevent the reassembling or coiling of double-stranded DNA or single-stranded DNA into a double-stranded DNA conversion. So this is the single-stranded binding protein. And when the initial melting gets started, we require the DNA gyrase, and this DNA gyrase, what to do? DNA gyrase, it is acting as a topoisomerase. In case of E. coli, there will be the type 2 topoisomerase is present. DNA topoisomerase introduce negative supercoiling. What to do? Introduce negative supercoiling. If we want to replicate all DNA, the DNA must have in the condition of negatively supercoiled. Negatively supercoiled DNA get replicate, and DNA G is acting as a primer to initiate the mechanism of initiation process. So this picture shows how the initiation get elongates. So in this picture, we have, this is the region. This region is converted like this region. So this is the 5' to 3' direction, and this will be the 3' to 5'. And as you all know that the replication always 5' to 3' direction only. So there will be the hexameric protein, what? DNA helicase. And if we want to replicate this strand, as you all know, DNA replication is a template dependent process. Template must be required to initiate the replication. So if the replication start this process, in the process 5' to 3' direction, 3' OH provide the polymerizing activity of DNA polymerase, and this strand form a loop. Why form a loop? Because this strand is acting as a lagging. There will be requirement of multiple DNA G, or you can say primer, and this is the leading strand. These leading and lagging strands are found in eukaryotic cell also. So this is the mechanism of initiation, and the DNA polymerase, like this is a DNA polymerase, move in this direction, and there will be another DNA polymerase moves in this direction. So DNA replication. So DNA replication is a process in which 5' to 3' polymerizing activity done, and this feature is also applicable in the transcription process. There will be also the 5' to 3' directions only. So let's forward the elongation process. So this will be the replication fork. The Y-shaped structure is called the replication fork. And previously we have DNA like this. After that DNA like this. And after the initiation, let me clean this. You can visualize more better. Okay. So initially we have DNA like this. After the initiation process, we have initial melting. So this is the DNA, and this DNA get initially melt. So this initially melted DNA get converted into like this condition. So this is called the replication bubble.